Gastrointestinal physiology, intestinal fluid and electrolyte transport, an in-depth discussion on hydration and balance in the digestive tract. Welcome to our podcast, where we delve into the fascinating world of gastrointestinal physiology. Today, we are exploring the vital topic of how our intestines handle fluids and electrolytes, a process that is fundamental to every aspect of digestion and crucial for maintaining the body's fluid balance. Let us begin by considering the sheer scale of fluid movement in the digestive tract. Every day, the small intestine is presented with around 8 to 9 liters of fluid. This impressive volume is not only from what we eat and drink, about 2 liters, but also from our own body's secretions, including saliva, gastric juice, bile, pancreatic juice, and intestinal fluids, which together contribute roughly 7 liters. Out of this total, the small intestine is responsible for absorbing about 7 to 8 liters. The colon then takes on the task of absorbing most of the 1 to 1.5 liters that remain, such that only about 100 milliliters are ultimately lost in the feces. This entire process is highly dynamic, with both secretion and absorption happening at the same time. The balance between these opposing forces determines whether the intestinal contents remain hydrated or become dehydrated. At the cellular level, the enterocyte, or intestinal epithelial cell, is the star of the show. These cells line the gut and are connected by tight junctions that control what moves between them. On their surface facing the gut lumen, and on the opposite side facing the bloodstream, they have different sets of transport proteins and channels that carefully regulate the movement of ions and water. Let us talk about sodium first. Sodium absorption is the main driving force for water absorption, as water naturally follows the movement of solutes. There are several mechanisms for sodium uptake in the intestine. One major route is through sodium and glucose co-transport. This mechanism is particularly active in the small intestine, especially the jejunum. Here, sodium moves into the cell alongside glucose, or with other nutrients like galactose and amino acids. This process relies on a sodium-potassium pump on the basolateral membrane, which keeps the sodium concentration low inside the cell. As sodium and glucose are absorbed, they create an osmotic gradient, encouraging water to follow. This is actually the physiological basis for oral rehydration therapy, which is so effective in treating dehydration from diarrhea. Another mechanism is the sodium-hydrogen exchange, found throughout the intestine but most prominent in the jejunum and ileum. Here, sodium enters the cell in exchange for hydrogen ions being secreted. Additionally, in the distal part of the colon, sodium can be absorbed through channels known as epithelial sodium channels, which are regulated by the hormone aldosterone. This helps the body conserve sodium, especially in the final stages of digestion. There is also a sodium chloride co-transport system, similar to what occurs in the kidney, present in both the small intestine and colon. Chloride absorption is often linked to the secretion of bicarbonate. On the apical membrane of enterocytes, chloride is absorbed in exchange for bicarbonate, which helps neutralize stomach acid and alkalinize the local environment. The bicarbonate used in this exchange is produced inside the cell from carbon dioxide and water, a reaction catalyzed by the enzyme carbonic anhydrase. Bicarbonate itself can be both secreted and absorbed in the intestine. The pancreas, for example, secretes a large amount of bicarbonate to neutralize gastric acid entering the small intestine. In the duodenum and jejunum, bicarbonate can also be absorbed, often in exchange for secreted chloride ions. Potassium movement in the intestine is primarily passive, following the flow of water and concentration gradients through the spaces between cells. However, in the colon, there is also active secretion of potassium. Potassium enters the cell via the sodium-potassium pump on the basolateral membrane and exits into the lumen through specific channels. This is a major way the body gets rid of excess potassium. Water transport, meanwhile, is always passive. It follows the osmotic gradients created by the active absorption of solutes such as sodium, glucose, and amino acids. Water can move through cells via special channels called aquaporins, or between cells through the junctions that connect them. Now, the critical point is the balance between absorption and secretion. By default, the intestine is geared towards net absorption, to conserve water and electrolytes. However, secretion is also essential. It lubricates the gut, dissolves nutrients for absorption, and helps flush out potentially harmful substances and bacteria. The cells located in the intestinal crypts are the primary source of secretion. Here, a transporter on the basolateral side accumulates chloride inside the cell, When activated by signals such as acetylcholine or vasoactive intestinal peptide, chloride channels open on the apical side, allowing chloride to flow into the lumen. This attracts sodium and water, resulting in fluid secretion. Clinically, this system can be disrupted. For example, the toxin produced by the bacterium responsible for cholera permanently activates the chloride channels, causing massive, life-threatening watery diarrhea. On the other hand, cystic fibrosis is caused by a genetic defect in these same channels, leading to thick, dehydrated secretions that can block pancreatic ducts and airways. The regulation of fluid and electrolyte transport in the intestine is tightly controlled by neural, hormonal, and local chemical signals. For instance, stimulation of the sympathetic nervous system increases absorption and decreases secretion, helping to conserve fluid during times of stress. In contrast, the parasympathetic nervous system decreases absorption and increases secretion, which is important during digestion. Hormones like aldosterone enhance sodium absorption in the colon, while other mediators such as vasoactive intestinal peptide and secretin promote secretion. Somatostatin acts to reduce secretion. Regionally, the jejunum is highly permeable and is the main site for nutrient-coupled sodium and water absorption. The ileum is less permeable but still absorbs a significant amount of fluid and is responsible for the uptake of bile salts and vitamin B12. The colon acts as the body's drying oven, absorbing the last portion of fluid and producing semi-solid feces. Its impressive capacity to absorb water is a crucial defense against dehydration, especially during episodes of diarrhea. In summary, intestinal fluid and electrolyte transport is a finely tuned, high-volume balancing act. The orchestrated work of cellular transporters, channels, and regulatory signals ensures that we absorb the water and electrolytes we need, while also using secretion as a tool for digestion and defense. Thank you for joining us on this deep dive into the marvels of gastrointestinal physiology. If you enjoyed this episode, please like, share, and subscribe to stay updated with more insights from the Everything Surgery podcast, where we continue to dissect the extraordinary.